Dr. Bryson is currently a research associate at the Greensmith Laboratory at the Institute of Neurology. At the Institute of Neurology's Neuroscience and Movement Disorders Unit. In 2002, he received a bachelor's degree from the University of Leeds, completed a master's in 2006, and a second master's of science in 2008, both at the University of Columbia. And in 2011, he received his PhD from King's College London. In 2014, he was the recipient of the UCL Early Career Neuroscience Prize, and he's here today to talk about his lab's recent recent developments in his laboratory. So,
well, thanks very much for the invitation to and share these latest results from our research projects. I hope you find them interesting. So basically this project employed a combination of optogenetics and stem cell based neuronal replacement in order to overcome muscle paralysis. So to begin with, I'll just give a very brief overview of the voluntary motor system. So typically motor signals originate in the primary motor cortex in the brain, where they're relayed by upper motor neurons onto spinal motor neurons in the spinal cord, and the signals are then relayed along peripheral nerves to the intended target muscle. However, there's a wide variety of different pathological conditions and injuries, such as stroke, traumatic brain injury, spinal cord injury, and in particular the focus of our research group, the neurodegenerative disease, motor neuron disease, or amyotrophic lateral sclerosis. So this results in the permanent blockade of motor signals, resulting in permanent paralysis of muscles. So essentially there's a dire need to develop new approaches to overcome muscle paralysis and to restore motor function. So one of the most promising recent approaches is to use stem cells to replace neurons that have been lost. However, so typically motor neurons in the spinal cord are located within extremely complicated topographically arranged circuits, and most of the developmental guidance cues that are responsible for determining this circuit formation are largely switched off in the adult CNS. So this makes it extremely problematic to be able to replace the motor neurons that have been lost within the spinal cord. So here you can see an example of an ALS model mouse in which many of the motor neurons in the spinal cord have already been lost. So this is largely responsible for the ensuing paralysis in this model. So, as I mentioned, it's very difficult to integrate new neurons within the CNS. However, quite groundbreaking research by Rob Branston's group in Canada demonstrated that it's possible to transplant stem cell-derived motor neurons into injured peripheral nerves, and these will then extend axons along the nerve and form de novo neuromuscular junctions with intended muscle targets. So this represents a way of circumventing the complex circuitry within the spinal cord, but because they're completely isolated from the descending cortical motor signals, you therefore need an artificial means of stimulating the transplanted neurons. So in this case they used electrical stimulation of the transplanted neurons. However, this comes with the severe disadvantage that when you electrically stimulate motor neurons, you recruit them in such a way that you recruit the fastest, most fatigable motor neurons. Therefore, the muscles rapidly become fatigued. So this makes it very difficult to use this in a chronic situation. So I'm sure most of you have heard of the new field of optogenetics. So this is essentially a novel way of controlling neuronal activity by artificially expressing channel rhodopsins within the neuronal membranes. So in our case, we employed the channel rhodopsin 2 gene, which is a gene originally from marine algae that responds to flashes of blue light, which results in pore opening and influx of sodium and potassium ions, resulting in neuronal depolarization. another groundbreaking study that preceded our work demonstrated that it's possible to transgenically express these channel rhodopsins in motor neurons in mice, and this makes it possible to stimulate the axons in the peripheral nerves to elicit very finely controlled motor functions. And perhaps even more importantly, when you use optical stimulation versus electrical stimulation, you're able to recruit motor neurons and motor units in the correct physiological order. So this enables you to avoid inducing rapid muscle fatigue. So that was the kind of background to our project. So we then set about devising a strategy combining aspects of the stem cell-based replacement of motor neurons with the ability to optogenetically stimulate these neurons. The first step in this was to generate a tailorized line of embryonic stem cells from which we could derive motor neurons that could fulfill the very specific functions for this project. And the next step was then to test in vivo whether we could restore muscle function using optical control. And perhaps right at the very end, I'll just mention what we think the ultimate clinical application of this could be. So, as I said, the first step was to develop the line of ES cells. This work was carried out by our collaborator Eva Lieberman at King's College. And Eva is an expert at genetically manipulating these embryonic stem cells. So we were able to express not only the channel rhodopsin gene, but also two other key genes, one of which, the CD14 gene, allows us to sort out motor neurons from these differentiated cells. So this cluster of cells is called an embryonic body, probably only about 30% of which is actually motor neurons. By using the magnetic sorting technique, we can purify the motor neurons to about 95% purity. So another essential gene that this line of stem cells also express is the glial-derived neurotrophic factor. So here you can see motor neurons that have been differentiated from the ES cell line, and in red within the cells, you can hopefully see accumulation of the GDNF protein. So this enables these motor neurons to secrete their own trophic support, and this turned out to be essential in order to enable them to survive long-term, both in vitro and in vivo. And this long-term survival is essential for their the maturation of their electrical firing properties. So having developed this stem cell line and characterized it in vitro, the next step was to develop a model in which we could test whether we were able to re-innovate paralyzed muscles. So the model we chose to use is a nerve ligation. So essentially we tied a suture very tightly around the nerve proximally, and this results in complete degeneration of all of the distal axons. However, a small minority of endogenous motor axons are actually able to regenerate through the ligation injury. So this results in a partial denervation model, which quite closely resembles the scenario in ALS patients who have not quite complete denervation. So after the ligation, we then engrafted the ES-derived motor neurons into discrete branches of the sciatic nerve in the leg of mice. And depending on which branch we injected them into, we could then potentially re-innovate very specific muscle targets that lie distally along that along that nerve. So I just have a very brief video of the actual engraftment process. So we use a dye called mild blue in order to be able to physically observe the cells being injected into the nerve branches. So hopefully you can just see a slight blue stain of the the nerve, indicating that the cells have been taken up into the nerve. So as a first step, we wanted to establish whether, first of all, we could see survival of the engrafted motor neurons. So this is actually 5 weeks after the graft. And if you look at these confocal images of the graft site, you can see the specific functional label of motor neurons, choline-acetyltransferase. So this denotes that the cells are actually motor neurons, and in fact they have an identical morphology to endogenous motor neurons in the spinal cord. And by staining, we could also demonstrate that they express the channel rhodopsin gene. Additionally, when we looked a bit further down the nerve, distally, we saw robust projection of many hundreds, if not thousands, of motor axons from these transplanted motor neurons. And quite importantly, these motor axons were myelinated by the endogenous Schwann cells in the nerve. And finally, when we looked in the distal muscle target, we could also observe a large number of neuromuscular junctions labeled with this alpha-bunga responsin marker. And you can quite clearly see the overlap of the transplanted motor neurons in green with the muscle end plates shown in red. So one slight downside is the observation of these characteristic sprouts emerging from the NMJs, which is a hallmark of inactivity of the motor neurons, which isn't very surprising because the transplanted cells were simply sitting in the nerve. They weren't receiving any stimulation from the brain, and they weren't receiving optical stimulation until right at the very end point of the experiment. But we quite conclusively demonstrated that it's possible to get robust reinnervation of these denervated muscles. So the next step was to observe whether we could optically stimulate and control the muscle function of these transplanted cells. So in our lab, we employ a technique called isometric muscle physiology, which essentially enables us to record the force produced by the distal tendon of the specific muscles. So here you can see on the right an actual trace of, so it's shown in red as the electrical stimulus delivered to the LED light source, which turns on the light. So these the short peaks were elicited a very short twitch contraction of the muscle. But when we delivered higher frequency bursts of light, we were able to induce a fused tetanic contraction of the muscle. So additionally, since these motor neurons, as with all motor neurons, it was possible to electrically stimulate them as well as to optically stimulate the engrafted cells. So this enabled us to compare the time it takes for the signal to elicit a muscle contraction. And as you can see, the timescale is almost identical, if not slightly faster, for the optical stimulation. And this analysis just demonstrates that the latency to the response from the muscle is identical with both optical and electrical stimulation. So, as I mentioned earlier, one of the huge drawbacks of electrical stimulation is that it rapidly induces muscle fatigue. So here you can see a perfect example of a train of electrical stimuli delivered over 3 minutes, and very rapidly the muscle force begins to decline. Whereas with optical stimulation, the amplitude of the muscle contraction at the start of the stimulation period is almost identical as at the end. So this confirms that optical stimulation is able to recruit motor units in the correct order and therefore avoids muscle fatigue. An additional extremely important finding from the physiology analysis was that by adjusting the intensity of the optical stimulation, we're able to recruit varying numbers of motor units. So so I should maybe just explain a motor unit is a single motor neuron and all of the muscle fibers that it contacts. So quite often with large motor neurons, they'll innervate up to 100 different muscle fibers. So each of the lines on this trace here, each of the stochastic jumps represents the recruitment of an additional motor unit, thereby resulting in a jump in the force output of the muscle. So this ability to recruit different numbers of motor units is, again, extremely important in being able to control muscles in a physiological manner and to induce a graded muscle contraction rather than spasmodic contraction. So just to summarize these results very quickly, so we quite conclusively demonstrated that it's possible to engraft stem cell-derived motor neurons into peripheral nerve branches and thereby induce robust reinnervation of specific muscle targets. And additionally, the muscle responses can be finely controlled using optical stimulation. And perhaps most importantly, the optical stimulation has significant advantages compared to electrical stimulation. So essentially, these results form the backbone of our recent paper published in Science. And as a result of this publication, we were then able to secure 2 separate research grants, one from the MNDA and the other from the French equivalent charity. So these are the next goals of the project: to improve the cellular aspects of the graft, additionally to test whether we can get survival of the graft in sod mice which model ALS, and to be able to optically stimulate them over a long time period. So we've shown that they can survive long term, up to 12 months in mice, and additionally they can also survive in the potentially toxic environment in the SOD1 mouse model of ALS. And we've just recently acquired a couple of LED laser, blue light lasers that we intend to use to chronically activate these neurons using an implantable fiber optic stimulator. And just very briefly, so we think the intended clinical application of this might be to control the phrenic nerve mediated contraction of the diaphragm muscle, which is largely responsible for breathing. So in ALS and spinal cord injury, the phrenic nerve can either become injured or undergo denervation. Therefore, we think that by transplanting these stem cell-derived motor neurons expressing channel rhodopsin, we can re-innervate the diaphragm and then use an implantable fiber optic and optical pacemaker in order to control diaphragm function and enable patients to breathe without the aid of mechanical ventilator. So I'd just like to thank in particular my boss Linda Greensmith and our collaborator Eva Libram at King's College, and additionally his postdoc Carolina, who were instrumental in developing the cells required for this project. And thanks very much for involving
So I forgot to mention at the beginning that after each talk, we're going to take 5 minutes of questions. So if anyone has any questions, I'll start to Bryson far away now.
So do you reckon that this could also be used in implanted in hearts as to replace the sort of standard pacemakers that are currently used?
So potentially, yes. So obviously there wouldn't be motor neurons that innervate the heart. But in fact, so the vagal nerve, I think, is some cholinergic neurons that are responsible for slowing down the heart rate. And yeah, there is actually a group interested in expressing channel rhodopsins to do exactly that in vagal nerves that innervate the heart.
Is there some sort of future plan for using this technique on maybe more complex systems? Like you mentioned, in the spinal cord, most of the signals that lead to differentiation of gone by adulthood? Do you have a plan for?
Yeah. So initially, so the reason we're particularly interested in the diaphragm to start with is because it's a relatively simple rhythmic contraction. That is probably one of the most essential motor functions in the body. But so as I mentioned, we can engraft the motor neurons into very fine branches that innervate discrete muscle groups. So potentially we could have a scenario where you could innervate lots of different muscles and have several different fiber optic stimulators. So it's purely a matter of developing the stimulator technology rather than the cell-based technology.
I was just thinking with Parkinson's, they've tried to transplant stem cells into the they've tried to derive substantial neurons for Parkinson's, and those ended up degenerating following a number of years or months, I don't remember.
Yeah.
Is that what you're trying to do for the second part then with the SOD1 mice?
Exactly,
To see
yeah.
whether the nerve,
So,
whether the
yeah,
neuron
that's
going to-
a real problem because the environment in ALS patients and SOD1 mice is known to be toxic to motor neurons. But we think that because they're transplanted outside of the spinal cord, because they express the GDNF gene, which is highly neuroprotective, we think that it's quite likely they'll be able to survive in this ectopic environment, and indeed we've shown that they can survive till quite late stage in these mice. So it looks quite promising so far.
Why is it the case that electrical stimuli causes muscle fatigue?
So it's due to reverse recruitment of motor units. So it's due to Henneman's size principle. So the larger the diameter of the motor axon, typically the more stimulus is required to recruit the motor axon. So the larger the diameter of the motor axon, also the more muscle fibers that it innervates. So typically during a voluntary muscle contraction, you start by recruiting the smaller diameter muscle fibers and the smaller, slower contracting muscle fibers. And then as you intend to increase the strength of the force, you then increase the stimulus onto the motor neurons, and then recruit gradually larger motor neurons, which results in gradually increased force. But with electrical stimulation, it's not known exactly why, but for some reason, larger diameter motor axons are much more sensitive to electrical stimulation, so they become activated first. And then as you increase the electrical force, you then recruit the smaller motor axons, which is the exact opposite of what happens during a normal muscle contraction. So basically, it's trying to elicit the strongest force first, and then you recruit gradually weaker ones, which is the opposite of what normally happens. And these strong muscle fibers are particularly sensitive to muscle fatigue.
Thank you very much.